We have some time for some questions. And so if people want to send a chat text to us, we'll pick out a few questions for Alex to ask. And our first question is from Bill, and he's asking, how did you get interested in the study of HIV/AIDS?
That's definitely a long journey where I initially got a summer job working in a crystallography lab where you try to figure out the structures, the atomic structures of protein. And that was before I started my undergraduate studies at the University of Missouri-Columbia, or Mizzou. And in his lab, I started learning about protein and some of his previous work solving the structure of interferon, which is used to help treat some cancer patients and hepatitis patients. And that really got me started in the idea of structure-based drug design. Then I joined Tom Quinn's lab, where we worked on the design and evaluation of potential compounds to image and treat cancers. And that got me started in the computer-aided drug design. When I went to grad school in Andy McCavin's lab at UCSD, that's when I really started working on HIV protease initially, and learned more about the AIDS crisis and the fact that it's one of the 3 deadliest infectious diseases. So HIV, malaria, and tuberculosis kill more people than any other infectious diseases, with HIV being the worst virus out there. And so because it kills so many people and causes so much human suffering, that's what really got me into doing science as a whole, as a career, and specifically to work on HIV.
Okay, thank you. We have another question from Richard. How would these compounds affect the production of regular cells in the body, which reproduce every 7 years or sooner?
So after we find these hits and then, spend years turning those into actual leads and then drugs, those drugs have to be tested against human cells as well as, the viral cells to make sure that they're really specific against the infected cells and don't kill the healthy cells. But then you do animal studies and trials in people in order to try to minimize those other side effects that are produced. But really you have to do the test in animals and then people in order to figure out whether they kill off the cells that they're not supposed to affect or not. So it really depends on each particular compound, and we'll have to take these hits and make them much more complex before they're potent enough to be a potential drug, and then those particular compounds have to be evaluated individually to see whether they cause too many side effects.
Okay, we have a question from Ronnie. What about entry inhibitors? They do not require a cell that would catch it on cell penetration, I guess.
Right. So those are some of the newer types of inhibitors like Fusion that target the fusion event of how HIV is able to bind and enter a T cell. Those are much newer. The ones that I'm aware of are, actually there's 2 different flavors of these entry inhibitors. There are some peptide-type drugs that target the viral spike and gum it up to prevent the conformational changes that it does during fusion. So those tend to be peptide or protein drugs, so you can't really turn that into a pill. Those can only be injected, so that limits the amount of people that can get it. A newer type of entry inhibitor is, I believe it's called Cell Sentry from Pfizer, that binds to the human receptor on the T cells that HIV exploits to enter and infect the cell. And in that case, those just bind to the human protein and block it so that HIV can't use that door. So we're not currently working on those particular targets. Right now we're focusing on protease and integrase. But once we've made enough progress against these targets, we can definitely move on to some other ones, such as the viral spikes or some of the other targets that are out there and that they're getting more data on.
Okay, Stephen would like to know if we find an AIDS drug with this project will be in the public domain or generic?
Right. Any data from the World Community Grid is in the public domain. So if any other scientist wants access to this data, they just have to ask us. Now, in general, these experiments are really focusing on finding hits. Sorry about that background phone there in a conference room. I wonder if I can mute that. No, sorry. Right, so any of the data that we find from FightAIDS at home is in the public domain. If people want it, they can access it. But on these experiments, it's very unlikely we're going to find something potent enough to be a real drug. We'll just find these hits, these scaffolds that one could then develop and make more complex into a drug. if we actually do find something potent enough to be a real drug in these experiments, and that would definitely be in the public domain, but in all likelihood, these compounds we discover here are going to have to be made much, much more complex. So we share the data from the experiments and any of the hits that are proven to be active in the test tube, so that many other labs, whether they're in other universities or research institutes like us, or in their company or whatever, they can use that data and then try to develop those hits into their own drugs, so that we have more and more new types of medicines coming out there.
Okay, another member is asking, is there any work being done with variable surface glycoproteins?
Not on FightAIDS at home itself, but the first postdoctoral fellowship I did in Professor Steve Mayo's lab at Caltech, the main project I worked on was exactly about targeting GP120, which is the main surface glycoprotein at the tip of the spike. The spikes are what's used to actually attach and infect the human cells. And so there we worked on the engineering of antiviral lectins to bind to those spikes and either form a cap on them and or gum it up to prevent the conformational changes required for the fusion event. But those are protein-based drugs, and that project is being continued back at Caltech. But here on FightAIDS at home, we're focusing on like the small little chemical compounds, the small molecule, and we're working on different targets because we need something with a really high-resolution, atomically detailed map of exactly what shape the target has, so that then we can simulate the different shapes it can sample. Whereas with these surface glycoproteins, there isn't that structural data that we need for our calculations. But in the future, we certainly could target those.
Okay, and another member wants to know if you've been able to create or advance the drug, or are you close, like 5 or 10 years?
It's really hard to estimate that. Thus far, a lot of the candidates that we've initially identified from FightAIDS at home that I've discussed in some of the FightAIDS at home newsletters, we found some that would kind of work a little bit, but they don't display the right type of signature for a real inhibitor, and that's like called a dose-response curve, that when you increase the amount of the compound, you increase the dose, it should actually inhibit the target more. So some of the ones we've discovered don't display that dose-response relationship, or when we test it with certain other types of compounds, it indicates that they're probably non-specific and whatnot. So thus far, we haven't discovered any great new hits, but we're certainly working on it, and a lot of these compounds do have the potential, and there's a lot of other data that we have yet to analyze that should hold some other promising candidates. But once we've shown that these candidates actually work by our collaborators doing those test tube assays, then we'll try to develop that into a hit and a lead. But thus far, we haven't found anything on FightAIDS at home itself, although in some of the other work I do as part of the Olson lab at Scripps, I'm working on some lead compounds discovered by some of the other collaborators, if I still have the pointer here. So some compounds that have been discovered in Valery Folkin's lab and MG Finn's lab, those weren't from FightAIDS at home, but they're part of our overall anti-AIDS team. And so I've been working on trying to develop some of those leads into something more potent. And also Stefano Forlì is taking some of the allosteric fragments that we discovered and is currently trying to turn those into more potent inhibitors as well. But at this point, we're still focusing on trying to find some new hits and then actually proving that they work. And those wet lab assays for our collaborators can take several months to a year just to figure out and show that it really does work. And then it's going to take us, at least a few years to try to turn those hits into something that's 1,000 to 1,000,000 times more potent before it can be a lead or a potential drug. Thus, if we had some really exciting new proof to share, you would have heard of it. But there's definitely a lot of promising data that we still have to go through and that we're still currently generating on a daily basis.
Okay, thanks. So we have about 7 more minutes of questions, and we're probably not going to get to all of them. Here we have another question. What are typical inhibitor constants you have obtained, order of magnitude?
Thus far, those non-specific inhibitors that didn't really pass through the wet lab test are in the micromolar range. So basically, they need to be nanomolar or picomolar range to actually be a lead or a drug. So we need to make it 1,000 to 1,000,000 times more potent. Now, some of the compounds that I'm working on that were discovered by our collaborators in MG Finn's lab and Valery Folkin's lab, those are already in the mid-nanomolar range, so I might only need to make those about 10 to 100 times more potent before they have potential.
We have one question about using GPU processing, and this project does not currently use GPU processors, but some of our other projects may in the future, so that's something to watch out for.
Right. I see a question from Mark Barsom about what needs to be done to apply these algorithms to other diseases, and we're already actually doing that. So the auto-dock code that we use to actually do the docking calculations to figure out whether compounds have potential to bind to a target or not, and how they would bind. So that's already shared freely. It's an open source program, so people can download that from the Olson Labs website at Scripps. Also, the protocols that we're developing and the new tools that we're making in order to speed up the ability to create these input files for the calculations, as well as to greatly speed up the ability to analyze the data and to make a more thorough analysis. So those tools are all shared freely with the entire scientific community. They just need to go to the website and download it, and we're trying to make people more aware of these great new tools that we have developed and are developing.
Okay, thanks. And Saul would like to know how would a drug be distributed if one was found, and how would it sell at low prices and so forth?
Right, so that's really outside of my area of domain. That gets into like the pharmaceutical companies and such. So we focus here on trying to find the hit, the new scaffolds or the new foundations for totally new types of drugs, and these new scaffolds can then be developed by many different labs in academia or at companies. If it's at a company, then they would certainly, if they produce a successful drug based upon these hits, the hits that we share with the public, then that particular drug would be a novel compound, and then they would, patent it. If you don't patent it, then no one will actually mass produce it, because they have to be able to get a return on their investment before they'll actually spend the money to mass produce it and distribute it to patients. But those aspects are really in the domain of the companies, and I'm definitely a pure academic researcher where I really just focus on coming up with new strategies and protocols and tools, and then trying to find these hits that many other labs can then try to develop to produce hopefully many different types of drugs.
Hey, Richard has a follow-on question. Why can't you use World Community Good to make some of those human protein interactions? This is probably in response to the original question he had.
Could you repeat that?
The original question was how would these compounds affect the production of regular cells in the body, which reproduce every 7 years or sooner?
Right, so it really depends on the exact particular details of any of those optimized leads or drugs. So you can't really predict in advance whether it's going to kill off those other cells that are normally reproducing instead of just targeting the HIV-infected cells. Now, there are other labs who entirely just focus on how to target drugs to infected cells, virally infected cells, or just the tumor cells and whatnot, and there are whole labs that just focus on that. But we really work on different aspects of the entire drug discovery process.
All right. Well, thank you very much, Alex. We've run out of time, so this has been a very informative discussion period here, and we, again, encourage everybody to go to worldcommunitygrid.org and join if you haven't already joined, and get your friends and relatives to join as well, because this is a great cause to help humanity in the world. So this concludes our webcast. Thank you very much.